This is the first part of the Chapter 3 lectures, recorded lectures on cells for anatomy and physiology. There are 4 sets of slides, and this set is part A. So I will try not to make this any longer than it has to be or needs to be. Excuse me. Here we go. Remember the video on why this matters, but I am going to skip that because we have quite a bit of material to cover. So you can look at that when you're reviewing the slides on their own. The first thing in section 3.1 is the cell theory, is making sure you know the components of the cell theory. The first component of the cell theory is the cell is the structural and functional unit of life. Oh my goodness, I didn't mean to do that, sorry. A cell is the basic unit of life, is another way to say that. The second component says how well the entire organism functions depends on individual and combined activities of all of its cells. And then the third component, structure and function are complementary, so that principle of complementarity, like we have with anatomy and physiology. Anatomy and physiology are complementary to each other. Is another way to say it, is structure and function are complementary. So the biochemical functions of cells are dictated by the shape of the cell and specific structures within the cell, subcellular structures. And then the continuity of life has a cellular basis. You may have learned it this way if you learned the cell theory in the past in other classes. Cells can arise only from pre-existing cells. So the only way we can get a new cell is when an old cell divides. Okay? There are over 250 different types of human cells, and the number of human cells is in the trillions. It's around 10 trillion, and that does not include the about 90 trillion microorganisms. Okay, we can see that human cells have a great amount of diversity. We see a picture here of some epithelial cells. This looks like cells that might form the kidney tubules because they're cuboidal in shape. We will talk about different tissues, and epithelial tissue is one of the tissues that we'll learn about. But these are cuboidal shaped epithelial cells. You can see the nucleus inside the cell, and the cell itself is shaped like a cube. Red blood cells or erythrocytes have a biconcave shape. That means they're concave on the top and then they're concave from underneath on the bottom as well. They almost look like a donut, except the hole has not formed all the way through. So they're very flat, rounded, flat cells that are concave on the top and bottom surface. And then we have smooth muscle cells and skeletal muscle cells, macrophages, which are very large white blood cells. You can tell down at the bottom, I'm sure you recognize the sperm because of its long flagella, its long tail. And a nerve cell is going to have lots of extensions and branches. A fat cell is going to be filled with fat. In a generalized cell, in human cells, there are 3 basic parts. There's the plasma membrane, which is the outer boundary of the cell. There's the cytoplasm, which is the fluid inside the cell that contains organelles, or little parts that help the cell to function. And then the nucleus contains the DNA, and it's also called the control center. If you look at a generalized cell, these are the organelles that you would typically find. Here is the nucleus, the largest organelle. Inside the nucleus is the nucleolus, and the DNA is spread out and uncondensed or uncoiled in a form we call chromatin. The nuclear envelope has these large pores in it called nuclear pores. And let's see, we have the plasma membrane on the outside of the cell. We have attached to the nucleus, we have the rough endoplasmic reticulum. You see all these dots that are attached to the outside of the rough endoplasmic reticulum. They're called ribosomes, and their job is to make proteins. Next to the rough endoplasmic reticulum is the smooth endoplasmic reticulum, which doesn't contain ribosomes. So its job is going to be to produce lipids primarily. We have the cytoplasm, which includes the cytosol, or the fluid, and it also includes all these organelles. This is a mitochondria, which is where cellular respiration occurs and ATP is generated for energy. Here's a lysosome, which is a vesicle that contains inside of it digestive enzymes that help break down not only the food that we eat, but also old and worn out organelles, germs that come into our body, old and worn out cells. So the lysosome can release these enzymes and digest pretty much anything. These centrioles help with cell division. The elements of the cytoskeletal elements are microtubules, intermediate filaments, and what are the other ones called? I'm sorry. I can't think. Okay, we'll come back to that. Then we have peroxisomes, which are similar to lysosomes, but they typically break down fatty acids. And they're called peroxisomes because they produce peroxide as a byproduct. Then we have the Golgi apparatus, which packages products from the endoplasmic reticulum, packages them into vesicles, and releases those products outside the cell in a process called exocytosis. Substances found outside cells we call extracellular materials. So that's going to be extracellular fluids or body fluids, such as interstitial fluid. Often this is called tissue fluid, just by regular language, but it's interstitial fluid or tissue fluid that surrounds our cells. And blood plasma is the fluid found in the blood that surrounds our blood cells. And then there's fluid that surrounds the brain and the spinal cord called cerebrospinal fluid, or CSF. And then we have saliva and mucus and other types of cellular secretions. And then the extracellular matrix is going to consist of substances that hold the cells together. So for the plasma membrane separates the intracellular fluid from the extracellular fluid. In other words, the fluid inside the cell from the fluid outside the cell. We also call it the cell membrane, the plasma membrane or the cell membrane. Now, the components of the plasma membrane are lipids. They form a lipid bilayer or a phospholipid bilayer. Within this phospholipid bilayer are proteins called membrane proteins, and they float through this lipid bilayer. And since they're constantly changing, and it's like the plasma membrane is made up of many pieces of these membrane proteins floating around in the lipid bilayer, we call it a fluid mosaic model or fluid mosaic pattern. On the surface of the plasma membrane are chains of glucose, which we call the glycocalyx. And then we have junctions that hold the cells together. They're called cell junctions. And you can see here the phospholipid bilayer is going to be these little round heads that have two tails attached. You have a top layer and a bottom layer. The tails are facing each other, and then the round heads are facing either toward the extracellular fluid or toward the intracellular fluid. But it's a double layer of these phospholipids, forms a phospholipid bilayer. These large purple molecules scattered within the phospholipid bilayer are the membrane proteins. They have various functions. And then we see these little green molecules that are shaped like hexagons. They are glucose. So chains of glucose or carbohydrate chains form the glycocalyx. We also have cholesterol. This is cholesterol that can be found within the phospholipid bilayer as well. Watch the animation. This goes along with slide number 14, the animation on membrane structure. You can see the phospholipid bilayer, but it's going to talk to you about the other components of the plasma membrane structure. All right, the lipids that compose the plasma membrane, 75% of those lipids are phospholipids, and a phospholipid has two parts. It has a phosphate head. Because the phosphate head is polar, it is also soluble in water, so we call it hydrophilic or water-loving. So the phosphate heads will face either outward toward the watery environment surrounding the cell or inward toward the watery environment inside the cell. Phosphate heads are polar and hydrophilic, and then the fatty acid tails face toward each other so they don't contact water. They are nonpolar, and since they are insoluble in water, we call them hydrophobic or water-hating. Another 5% of the lipids are called glycolipids, and these are phospholipids that have that carbohydrate chain on the outer surface. They're part of the glycocalyx. And then we have 20% of the lipids are cholesterol molecules that increase the stability of the plasma membrane. Here is your polar hydrophilic phosphate head, and here are your nonpolar hydrophobic fatty acid tails. And now we're going to talk about the membrane proteins. Membrane proteins allow cell communication with the environment. They have many functions. There are two types, integral and peripheral. Integral means that they span the entire plasma membrane, and peripheral means that they're only on one side, outside or inside the membrane. Okay, so this shows you all the membrane proteins. There are transport proteins. There are receptor proteins, enzymatic proteins, cell junction proteins are down here at the bottom. Cell recognition proteins, like you see with tissue compatibility, when you're trying to find a tissue match. These are proteins that are specific to individuals. Your cell recognition proteins are probably much different from mine, for example. And then we have attachment proteins, okay? So starting with transport proteins, they can be just channel proteins that allow a substance to just be transported across the membrane, either from outside to inside or from inside to outside. Then you can have proteins that we call carrier proteins that actually change shape whenever they are transporting. They bind to the substance and change shape and then transport it through the plasma membrane. So transport proteins literally transport some type of substance across the plasma membrane from outside to inside or inside to outside. Some of them require ATP and some don't. There's a good animation in slide number 22 on transport proteins. All right, I'm trying. Here we go. All right, then we have receptor proteins. Chemical messengers like insulin or other hormones will bind to the receptor, and then that will initiate some type of change inside the cell. If it were insulin binding to the protein, it would cause that cell, possibly a liver cell or a muscle or a fat cell, to take in glucose from your blood